Hello everyone, welcome to today's webinar. I'm Mitch Andre Garcia, Branded Content Editor for CNEN, and I'll be moderating today's event. This webinar is titled Breakthrough in Nanoparticle Tracking Analysis and is being sponsored by HORIBA. During the presentation, we will hear from Rick Cooper, who is the Chief Executive Officer of MANTA Instruments. Rick is a serial entrepreneur with over 20 years experience in senior management positions and leadership roles in 8 startups. For over 30 years, Rick was involved with cutting-edge electrochemical device development, including pioneering work in rechargeable lithium batteries and fuel cells used in multiple industries, including automotive and consumer electronics. He's an advisor at the Von Liebig Entrepreneurism Center at UCSD, former treasurer and board member of Tech Coast Angels, a Southern California-based angel investment network. Rick has honors degrees in chemistry from the British Columbia Institute of Technology and Mechanical Engineering from California Polytechnic University, San Luis Obispo. I will now hand over to Rick.
Hey, Mitch. Thanks for the intro, and I'd also like to thank everyone for joining us here today. Really appreciate your time. Again, I'm Rick Cooper, CEO of MANTA Instruments. At MANTA, we're producing scientific instruments that measure the size and concentration of nanoparticles more effectively than any of the available products. Our product is based on breakthrough technology developed at UCSD, where researchers there have been working with nanoparticles for over 30 years. And they have raised millions of dollars in grant money purchasing all of the best available instruments. During this extensive career in nanoparticles, they were routinely frustrated with the limitations of the existing technologies. And as they say, necessity is the mother of invention. So good for them. They didn't just sit on their hands. They went out and got a separate research grant for scientific instrument development. And after a 3-year R&D effort, they came up with the breakthrough technologies that are used in MANTA's products today. So with that background, I will move on into the presentation. Today, we'll cover some basic background, just reviewing some of the existing products, get into the details of our technology. The vast majority of this presentation includes tests from experiments that we've done at MANTA and that we've done with other customers and partners. Hopefully you'll find something that's comparable to your application. If not, we have many other tests and results that we can share with you if you're interested. And the end of the presentation will include the specifications for the product and other details on our product. So here's a summary of the established technologies that researchers at UCSD and others have been using for a very long time. Dynamic light scattering, typically referred to as DLS. What we call conventional nanoparticle tracking analysis, or NTA. We call it conventional since our technology is an advanced version of NTA. And of course, you have electron microscopy and flow cytometry as other techniques that are used for working with various sized particles in the nanoparticle range and micron size range. Those technologies have these fairly well-known unmet needs, particularly when it comes to polydisperse samples. The ability to visualize a wide range of particles is not easy. And then accurate and reproducible measurements of number, concentration, size distributions, and kinetic processes, particularly when you have different ranges of sizes of so-called polydisperse samples, are, as of today, or let's say until MANTA's product came into the market, were unmet needs. The goal of my presentation today will be to show you how MANTA has addressed these unmet needs with one simple-to-use benchtop instrument. So let's move on to the technology and how it works. Like DLS and conventional NTA, we're using a light scattering method. We do this because the particles themselves are similar to or smaller than the wavelengths of visible light, so you literally cannot use any optical microscopy for this type of particle size range. However, you can rely on images of scattered light from the particles themselves. And it's very well known that these nanoparticles are exhibiting Brownian motion, and their particle movements are directly related to their size. We capture the motion on video recording equipment, and from the motion, we can accurately calculate the size. This diagram is oversimplified, but we're showing here that we're doing this on an individual particle basis. So particle by particle, we're tracking particle motion, and therefore we can count each particle. And because the volume that we're examining is very well known, we can determine the concentration of the particles in the sample. And we can do that for different sizes in the same particle, in the same sample. Here we have just a little animated GIF showing how the smaller particles always move more vigorously than the larger particles due to this Brownian motion effect. So, and they're just basically diffusion effects that cause the different motions. So this all sounds nice and easy, pretty straightforward. However, there's a significant and fundamental problem with any light scattering technology. And that basically boils down to this huge dynamic range of scattered light intensity as a function of particle size. Here we're plotting angular scattering coefficient as a function of particle diameter, and the coefficient is directly related to the scattered light intensity. So for the typical range of interest from 10 nanometers to 1,000 nanometers, 2 orders of magnitude of particle size, you typically have 8 orders of magnitude of scattered light intensity. This causes havoc for the detection system in scattered light systems, whereby the larger particles are just scattering so much light, it becomes very difficult to observe and get any meaningful information from smaller particles when larger particles are also present in the sample. These problems have well-known effects for the other light scattering methods. In dynamic light scattering, it's common that the results will be skewed towards larger particles. And it's not uncommon that a few contaminants, a few pieces of dust or whatever, could literally render the results meaningless. In conventional NTA, differently sized particles in the same sample cannot be seen, and therefore if you can't see them, you can't count them, you can't size them. As I say, if you just need to be able to see the particles before you can effectively measure them. This problem, as I said, is very well known. As a matter of fact, there was a recent standard for this method that they call particle tracking analysis. Most people call it nanoparticle tracking analysis, but ISO has characterized it as particle tracking analysis. In their recently published standard, they included these remarks about the complications of polydisperse samples. So polydispersity affects the ability to track and therefore analyze different size fractions. In a polydisperse sample, large particles scatter a lot more than small particles, making it difficult to detect or track small particles. So that's pretty good validation of this well-known problem that we've talked about at the beginning of the presentation. Let's contrast that with a reference customer from MANTA. This is Dr. Sadek Esner. He's director of the Cancer Early Detection Research Center at OHSU. And here's what he has to say about the ViewSizer 3000, which he purchased from MANTA. We confirmed this easy-to-use benchtop instrument meets all our needs for visualizing, sizing, and counting nanoparticles, and you can see the range of different types of particles that he's working with. And this is the first product we found that can effectively characterize particles in polydisperse samples. I also like to highlight the concluding phrase here where he's talking about the elements of mystery associated with other methods. Some of you may have these same kind of feelings where you've put a sample into an instrument and you've been able to get some data, but because you can't actually visualize or see the sample, you're kind of wondering if the results are true or if they're accurate, or how do you really get some kind of confidence that the results are accurate. We find this visualization capability provides a tremendous degree of comfort for scientists working with nanoparticles, because you can literally see images of the particles and you can sit there and actually count them on the screen if you're interested in some degree of validation. So yeah, we're pretty pleased with his feedback. So you may be wondering, how do we do this? Happy to share the details here. As is the case in many technology breakthroughs, it boils down to breaking the problem into manageable segments. At MANTA, we do that with something that we call simultaneous multispectral particle analysis. So we illuminate the sample with 3 different wavelengths of light simultaneously. This diagram is an oversimplification. There's no separation of the different illumination zones. As a matter of fact, there's complete overlap. All particles are illuminated with 3 different wavelengths, and the detection system can separate those 3 different signals of scattered light from each particle, and the software can choose the best image for each particle to allow us to pick the best particle for the tracking analysis that allows us to size them and count them. So this diagram is basically intended to show that typically blue is better for smaller, green for mid-size, and red for larger. But again, this is a simplification. All particles are illuminated with all wavelengths. People often ask, what does MANTA stand for? So I'll answer that one now. It's Most Advanced Nanoparticle Tracking Analysis, based on this simultaneous multispectral particle analysis. So the proof is in the pudding. This is a video from our instrument showing a very wide range of nanoparticles being examined simultaneously. In case the video doesn't work well for all of the viewers on different applications, I've captured a screenshot from the video here, so you can see a still image of all of the different particles that we were able to view simultaneously in the same video, in the same sample. In this case, we're observing 80, 240, and 700 nanometers very effectively. You can see these beautiful, clear, crisp, sharp images of scattered light from each particle in this mixture. It's always fun to go out and show these capabilities to different customers, and I'm pleased to say that we get a lot of great feedback from customers. Here's some unsolicited comments from user experiences when they first get to test their particles in our instrument. Wow, amazing, OMG, can't do my research without MANTA. The others can't do this. And are you sure that's the right price? I'm happy to report that's in the right direction. People are usually thinking the price would be higher given all the capabilities that we provide. Okay, so let's move on to the fun stuff. As I said, most of this presentation are data and results from a number of experiments that we've done. Just to reiterate, I can't show everything that we've done. There's been dozens of different tests. I think this captures many of the most common examples. And as I said, I'd be very happy to engage with anybody separately if they're interested in learning more about how their application may work with this product. So let's start with the bread and butter. This is the actual histogram produced from the measurement of that polydisperse video, which I showed a few slides ago. For all of these histograms, there's a few of them in the report, we are plotting particle concentration as a function of particle diameter. So again, to reiterate, this technology is able to measure particle concentrations very effectively, and it does it for different constituents in the same sample across a very wide range of sizes, as we can see from this particular experiment. These were very well-known polystyrene standards of known size, and we accurately determined the size. The concentrations were validated independently from a sophisticated spectrophotometric method that confirmed the concentrations were also accurate. So if you can do a complex polydisperse sample like that, then monodisperse is very easy. Won't spend a lot of time on this particular slide, but again, just to show you we're plotting concentration as a function of size. And this is an indication of the lower detection limit for metallic particles. These happen to be gold particles. Now back over to a polydisperse sample, just to show you that the technology does not depend on the type of material that's being analyzed. As a matter of fact, it's completely material independent. We have two different types of materials being tested here in the same sample. We had 30 nanometer gold particles mixed together with 400 nanometer polystyrene standards. And again, you see this very good separation across a wide range of sizes and independent of particle type. Before moving on to the next series of experiments, I'd just like to highlight that this capability to visualize size and count wide-ranging sizes of particles in the same mixture opens up a fascinating new field of nanoparticle process analysis, what we call kinetic analysis. So we can literally watch these smaller particles become larger through maybe, not for these particular materials, for other materials, through maybe a crystallization reaction, or perhaps aggregation of particles can be visualized and quantified over time. And then similarly in the other direction, you can watch larger particles become smaller, say for example, in a dissolution experiment. And that's the next example that I will show. So this is a crystalline API from a very well-known pharma company that's being developed, and it's designed to have a particular dissolution rate for its effectiveness. So we're starting off here with a high concentration of the particular API particles in solution. And over time, in this case it was around a 40-minute experiment, we watched these particles dissolve into solution. So we don't have 40 minutes to show the whole video. I'm going to show you a series of slices from the video as these particles are dissolving. So you get the feeling of the visualization of a dissolution experiment. It's a little bit choppy because there's slices, but I believe you'll get the effect. At the conclusion of the video, I'll come back to this frame so you can see again the concentration at the starting point is much higher than the end when most of the particles are dissolved. So here we go with the video. Particles dissolving over time. And I will just jump back to the first frame again as a reminder of the starting concentration, just so you can see the effect of start to end and then back to the start again. This visualization of particle concentration and size changes over time allow us to come up with elegant representations of the dissolution rate constants for experiments like this. So this is just one example, and we're told by the researchers that are doing these kinds of tests that their best alternative for this method is to somehow go back and forth between some dissolution apparatus and an HPLC to measure chemical uptake as the particles are dissolving. There's a number of lags in that process, and delays, which makes it very hard to get that steep part of the initial part of this curve. People in the pharma space and other places that are working on dissolution experiments are excited about this capability to visualize dissolution for particles in the subvisible range. So, as I mentioned, we can watch particles dissolve, and we can similarly watch particles crystallize or grow in various mechanisms. This particular experiment is a combination of a crystallization and a dissolution experiment with the same product in the same sample continuously observed with the ViewSizer 3000 from MANTA. So at the top here, I'm highlighting the crystallization phase, which occurred over a period of approximately 430 minutes. And the initial particle sizing is done in what we call the Brownian motion mode, and that's the method I showed earlier, where we're observing particles exhibiting Brownian motion and looking at their size from diffusion expressions. Once particles become a certain size, typically around a micron, the Brownian motion becomes de minimis, and it comes to a certain size, and those particles are large enough to settle due to gravity. So we start observing them in what we call the sinking sizing mode. That's the zone in blue there with a little bit more scatter in the data, but nevertheless getting good representation of the continuation of the crystallization process. At around 430 minutes, the customer added PBS to arrest the crystallization, and then 450 minutes added additional PBS to begin a dissolution process on, again, exactly the same sample that was in the same tester through the whole process here. This particular sample happened to be a protein therapeutic that's being designed specifically in crystal form for more effective administration purposes. If you're interested in more info on that particular experiment, there's a recent publication in the March issue of Bioprocess International with significantly additional data and other information about the process. We're very pleased that this customer concluded that the results from these experiments enable them to do more effective material formulations as well as optimize their processes due to these results of measuring the process of crystallization and dissolution in real time. Here we're going to show the sinking effect. So if you look in the top right quadrant here, you'll see one particle that's moving around randomly more or less in the same spot. And above that particle, you'll see one larger particle that's essentially just coming straight down in the image. I'll just play that again quickly here. So again, that particle coming in from the top, we call sinking. The other smaller particle below it in Brownian motion. In this still image, which hopefully is clear for everyone, we're looking at superimposed frames from that video showing how the particles are translating over time. And again, in that top right quadrant, you see the smaller particles, more or less random Brownian motion that's being tracked, and then the larger particle is tracking mostly in the downward direction due to gravity-based settling. So of course there are diffusion expressions for this type of particle movement as well, and we use those diffusion expressions for the larger particles while simultaneously also measuring the size accurately for smaller particles. In this case, the Brownian motion particles were on the order of 100 microns, 100 nanometers, and the larger settling particles were on the order of a micron. So this massively extends our range of sizes we can work with In the crystallization experiment I showed a minute ago, we're doing this with particles up to 15 microns. There's no reason why we can't go larger. We report the upper limit so far as 15 microns only because that's all we've had time to measure so far. So I highlight in this slide as well the direction of gravity with the G. That's important to know because competing NTA technologies are observing in the direction of gravity and physically would not be able to make these measurements of particles settling. The particles would just be going out of focus as they transition from higher to lower in the field of view, whereby we're observing perpendicular to gravity, which allows us to get these settling measurements, which has many beneficial uses, including this one, which I talked about from the crystallization experiment, but also for some stability experiments. People are interested to know what these settling rates are and at what size settling starts, all things that we can do with the ViewSizer. So another example of kinetic processes that are getting a lot of attention these days are protein aggregation in the field of biologic therapeutics. This recent paper from the FDA highlights the importance of this area of aggregation. In this paper, they studied a product that was launched last year. Sadly, after launching, it was pulled from the market when a number of people came down with autoimmune responses, and sadly, some of them actually died from the treatment. It's the paper doesn't directly connect all the dots, but it talks about the increased levels of subvisible particles in the deployed version of the product, which was in a multi-use vial, compared to the single-use vial, which had lower particle counts. And the single-use vial was what they used mostly for the FDA trials. So again, aggregates in protein biologics are increasingly important and a big deal, particularly as people try to develop higher and higher loaded product for more and more efficacious products. This next video is literally visualizing protein aggregates in a neat biologic product directly from the syringe. This is a very complicated sample. It's highly loaded. There's silicon oil droplets, excipients, and the proteins themselves, the molecules in solution themselves, are typically have somewhat fluorescence, and there's an awful lot of background noise that you have to cut through in order to be able to get in and look at the actual aggregate particles themselves. In our case, not only do we have 3 wavelengths of light, but each of those lasers has individually adjustable power levels, which gives multiple degrees of freedom for the user to cut through background and other sources of noise to get in and look at the specific particles of interest. In this case, protein aggregates in the neat biologic directly out of the syringe. I should also mention that the product has onboard stirring. We use that for a number of applications. But in the case of evaluating protein aggregation, you can use stirring to look at the effects of agitation on product stability. We also have the capability to adjust temperature over a wide range, and we can program temperature ramps. Temperature is known to be related to aggregation. Furthermore, this is essentially an experiment done in real time in a cuvette. Therefore, it's easy to add some reagents or some potential contaminants or other ingredients that you're interested in understanding the effect on the aggregation event. And all of that can be done in real time. The next chart is a results of an experiment looking at the effect of temperature on aggregation, and clearly there was a strong relationship between the amount of aggregation and the temperature of the experiment. So those are the summary of kinetic processes. As I mentioned, there are several others that we have done, and we'd be happy to talk with anybody that's interested. This page summarizes all of the different types of materials that we've worked with so far. As I mentioned, the technology is sample independent. The material is irrelevant to the method, so long as there's a reasonable difference between the refractive index of the material and the liquid it's suspended in. So I certainly won't read through all of these, but almost 40 samples successfully tested so far. Final stage of the presentation is on the product itself. Our first product is the ViewSizer 3000. Here's a picture. It's a benchtop scientific instrument. Fits on any benchtop. One person can pick it up. And I'd just like to draw your attention to the little black knob on the front of the instrument. That's essentially the handle for the door. The operator opens the door, inserts the cuvette, closes the door, and from that point on, all of the interfaces with the software, and our software is designed to be user-friendly, simple, easy to use. With just a few clicks, you can be looking at these beautiful images of nanoparticles in your sample and then analyzing those particles within minutes. Very important to highlight that this is elegant technology. There are only 3 inputs required. Of course, you have to put the sample in. As I've mentioned, our sample is held in a cuvette. Cuvette provides all kinds of benefits. It's simple, it's easy to use, it's reusable, and you can do some real-time changes to the sample in the experiment, as we talked about before. Temperature is one of the important parameters. It's controlled accurately to the customer's set point. Viscosity is the final input. It's typically available readily from the literature. However, some people are working with mixtures of liquids, or they've got complex samples where viscosity may not be well known. In that case, you can actually use the ViewSizer to measure viscosity. Simply put in a known particle size with an assumed viscosity and measure the particle size. The difference between the measured size and the actual size is directly related to the difference between the measured viscosity and the estimated viscosity, or pardon me, the actual viscosity and the estimated or the assumed viscosity. So most importantly, again, it's an absolute method. No calibration standards required. Only these 3 inputs. Here are the specifications. The measured range of sizes so far is now down to 8 nanometers, up to 15 microns. As I said, there's no fundamental reason why we couldn't go to larger sizes with the settling experiments. The minimum sample volume is 0.4 milliliters. The actual examined volume is on the order of 2.5 nanoliters. So there's no reason why we can't go to much lower sample volumes. And I know in some cases customers are concerned about the amount of sample that gets consumed in a particular test. We're working in improvements to the cuvette that would provide smaller volumes for those folks that would like smaller volumes. That's not far off. And I'd like to highlight that the sample can actually be reused multiple times in the same cuvette for multiple tests. So there's some sample conservation capabilities with this product. So for example, we were able to measure crystallization and dissolution with exactly the same amount of sample in the same test. There we have the typical sample concentration range from 5 times 10 to the 6 up to 2 times 10 to the 8 particles per mil. It's important to understand this is what we call typical sample concentration. There's no reason why you can't measure lower concentrations than this. You just need to take more time, collect more data. It's just a matter of statistics to get good results for significantly lower concentrations below that. And at the high end, you can do all kinds of useful experiments with higher concentrated samples, for example, some of this dissolution work, but the actual starting concentration may not be as accurate because you'll have some particles overlapping. So you can just imagine in a highly densely loaded sample, thinking back to some of those videos, if one particle is behind the other, you wouldn't be counting that particle. So, but these are the typical ranges that we recommend for measurements, but there's capabilities to work outside of this range. Temperature control accurately with the instrument itself with a standard product over a wide range from 5 degrees to 50 degrees C. For those that have special needs outside of that range, there's an upgrade available to -15 up to 110 degrees C. As I mentioned, benchtop instrument, one person can pick it up and it operates in any typical laboratory operating environment. So kind of back full circle to where we started. Hopefully we've done a reasonable job of demonstrating that MANTA instruments with our first product, the ViewSizer 3000, is doing a great job of addressing the previous unmet needs, particularly for polydisperse samples when it comes to particle visualization, concentration measurements, size distribution measurements, and this really fascinating new field of particle kinetic processes that we believe that we have opened up. So in summary, I'm happy to say that we have this new breakthrough technology available in the market now since 2016. It's providing new and better particle characterization for our customers, and I'm very happy to say that the customers themselves, our customers and our partners, are very pleased with the results that we've provided for them so far. And also pleased to announce that recently we did establish a new partnership with HORIBA Instruments as our distributor for North and South America and recently expanding into Europe. This allows a newer company with a new technology like MANTA's ViewSizer 3000 to provide all the background and backup and support of a well-established company with decades of experience selling and marketing and supporting particle characterization instruments. We couldn't be happier about this partnership, and if anybody's interested in any of the commercial details of our product, HORIBA would be our contact partner for the U.S., North America, South America, and European markets. So I'd just really like to thank everyone again for joining us today. I'll leave this last slide up here on the screen. We may not have a chance to get to everybody's questions, or if some of you would like to ask questions directly, I'm available. I'll make myself readily available for anybody that's interested for a follow-up. You have my email and phone number here on the screen for any of you that may want to follow up with me. Thank you very much.